See, once they'll divide and decide on 1,000 fates. Did you ever figure how they know? Be Mac. We are built of modules combined in a planned out way. Each new piece must be told where to go. Oh, now there's a science helping us to understand how our cells encode this architectural plan, signaling each other with genetic tools. Oh yeah, wow. Phenotype the interface for mouse and man, genotype the files and the subprograms. What then are the switches, circuit boards, and boot code? Evo Devo, looking at the logic in the ways that we grow. Every gene directed by a signal key code. Proteins that can activate, enhance, or veto. Evo Devo, the signals are controlled by other genes that signal. Calculating in a network labyrinth, though, where the heart and liver and the hands and feet go. Signal mapping tells each region what it ought to be, yo. With circuits so deep they build up on. They're older than the paleo. The paleo's a wicked baby. In a crucial pathway, changes tend to get torpedoed. Where they go, calamity goes. As the cyclonic shape knows. See down, they cascade like a domino. Like you and I, Drosophila. The path that makes us optical was laid a long, long time ago. Back before we blew up the Cambrian like a bomb. Now my eye protein can make you see out of your bomb. And hedgehog and its relatives like Indian and Sonic. Set up in a gradient on segments embryonic. Split four brains and asymmetric parts depend upon it. Flipping on genetic switches and logic.
A revolutionary new technology now entering the stage: CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. The CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell: microorganisms, plants, animals, or humans. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. The means to edit the genome of a human embryo already exist, though the technology is still in its early stages. Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. As powerful as CRISPR is, and it is, it's not infallible yet. Wrong edits still happen, as well as unknown errors that could occur anywhere in the DNA and might go unnoticed. The gene edit might achieve the desired result, disabling a disease, but also might accidentally trigger unwanted changes. We just don't know enough yet about the complex interplay of our genes to avoid unpredictable consequences. Working on accuracy and monitoring methods is a major concern as the first human trials begin. And since we've discussed a possible positive future, there are darker visions too. Imagine what a state like North Korea could do if they embraced genetic engineering. Could a state cement its rule forever by forcing gene editing on their subjects? What would stop a totalitarian regime from engineering an army of modified super soldiers? It is doable in theory. While this might be a tempting reason to ban genetic editing and related research, that would certainly be a mistake. Banning human genetic engineering would only lead to the science wandering off to a place with jurisdiction and rules that we're uncomfortable with. Only by participating can we make sure that further research is guided by caution, reason, oversight, and transparency.
Josiah Zahner thinks everyone should be able to change their DNA. Josiah is a very controversial figure. He's got a scientific background with a PhD in biophysics, and he spent 2 years as a research fellow at NASA. But he left to start a company called the Odin. He sells kits to help anyone modify organisms for as cheap as $30. That's because he wants genetic engineering to be decentralized, in the hands and homes of everyone.
I think everybody's worried about the risks of genetic engineering and how it's going to hurt people, and nobody's worried about the risks of what's going to happen if we don't allow people to do genetic engineering. Because the problem isn't going to be like, oh man, that person has way too many enhancements or doesn't. The problem is going to be, is it an equal playing field? Does everybody have access to this technology?
And it's becoming possible because of CRISPR, a gene editing technology that can find and cut a sequence of DNA in a cell and replace it with another.
If it wasn't for CRISPR, I don't think my company would be possible.
Leading researchers say the technology is still too new to use on humans without potentially harmful consequences, and that the way Josiah promotes it is reckless. He's gotten a lot of media attention for his very public DIY stunts, like when he injected himself with CRISPR while giving a talk at a synthetic biology conference.
This will modify my muscle genes to give me bigger muscles. Did that just quiet everybody? Sorry.
The month after Josiah's injection, the FDA issued a warning that, for safety reasons, it's illegal to sell gene therapy products for use on humans. Despite the warnings, the Odin has sold tens of thousands of CRISPR experiments. You can't make an effective human gene therapy from just what's in these kits. You'd need more skills and supplies.
If you don't purify it properly, you're just basically injecting yourself with something that can cause an immune reaction. The majority of people probably won't kill them, but that's just the majority of people.
Josiah's public CRISPR injection doesn't seem to have done much to him. He doesn't regret doing it, but says he was surprised by the reaction he got in the months that followed.
Alright, so I'm going to try to shoot to do half of it on one side of my stomach and then the other half on the other side.
After I injected myself with CRISPR, the next biohacker to inject themselves was like a month later. I thought it would be years. You never want your actions to have a negative impact on people.
To so-called germline editing, right? So where you're really editing embryo or the gametes. It's really, really dangerous, right? If we cannot control the editing outcome, we're basically passing whatever the failed experiments to the next generation. In a species as a whole, human being species, this is something we don't want to do if we cannot do it safely. But nothing was more perturbing when, at the end of last year when news came, right? So there was a real editing experiment in embryo that causes done by He Jiankui, a scientist in China. Essentially, this led to the actual birth of a twin CRISPR baby, right? So something that people kind of feared, and the scientific community has been self-policing to avoid doing, and someone just pushed the envelope and did it all in once, right? So that was very perturbing, causes an outcry in the scientific community.
Engineer, Ginkgo, as a company that's doing genetic engineering, we kind of look to this sort of misquoting of Arthur C. Clarke saying any sufficiently advanced technology is indistinguishable from nature. Motivating? So it's certainly brought a lot of engineers to the field. That's what synthetic biology is. That's what iGEM is. It's a lot of engineers kind of thinking about, can I program bacteria the way I program computers? What would that mean? What would that mean for what we can do? And what would that mean for the way that we think about technology? If you can design bacteria, what could you do? Could you imagine bacteria that live in the soil and live on the roots of plants and provide nitrogen fertilizer to them so you don't have to add chemical fertilizers? There's already bacteria that do this for plants like soybeans and other legumes, but we can't do that with other cereal crops like wheat or rice or corn. So Ginkgo and Bayer have a joint venture, another company called Join Bio, working to engineer the bacteria that live in soil. And
Hey, Coppertop, was wondering if you heard about the Bayer. Yeah, the aspirin. Oh, yeah, I heard about this. These fuckers are buying Anthony Monsanto and changing their name to Bayer to avoid the negativity that comes with the Monsanto name, but not changing any of the fucked up practices like GMO seeds and hiding from the public how they are poisoning our food supply, etc. Yeah, how come this never comes up in the political debates? They never talk about it because these guys have so much. We will fucking murder you money if you talk to us, talk about us. As if this wasn't bad enough, I read up on Bayer and come to find out a few years ago these bastards supplied blood to hemophiliacs who needed blood transfusions and their products and had the AIDS virus in it and continued to sell it after they knew about it. They even admitted to it? Come on. Where did you read this? What in the fuck? How are these corporate fuckheads always allowed to get away with this unbelievable level of greed and soulless, inhuman behavior? So it wouldn't surprise me. This is getting really dark, people. All right.
At the effects of that on the gut and on the pancreas. And these are direct effects. What you have is the Bt toxin will bind to your gut, will go through the gut wall, get into the circulation, and will go right to the pancreas, and then it will affect it directly. They asked me, would you eat it? I said, no, certainly not the GM potatoes we worked on. If I could avoid it, I would certainly avoid it. That the animal that has been fed on GM has been changed. Now, when you are eating that animal fed on GM, you will be eating a different animal.
People had studied what are called quantitative loci in genetics, where you have something like the shape of the solidity of tomatoes governed by about 7 different genes. People study these things. They're called quantitative trait loci, TRAIT, QTLs. This is a branch of genetics where they try to measure the amount of contribution of given genes are to different things. It's now being applied, but it's been studied in plants a great deal.
If a tomato is genetically engineered with a fish gene, is it still vegan? My name is Pat Thomas, and I'm from the group Beyond GM. Most people, when they think about genetic modification, they think of this science-y, techy thing that's beyond the comprehension of most of us. What we try to do at Beyond GM is to make it understandable. What you find is that the need is really led by the market. The market wants to produce more cheaply. This is a salmon into which eel genes and certain growth hormones have been spliced so that it will grow twice as fast as other salmon. And what that means is our supermarkets and our restaurants have more salmon, more times of year for more people. We're bringing together chefs from all around the world for an amazing week of events around the Chef's Manifesto. There'll be a huge space in which you can ask questions and find out more about sustainability and how you can get involved in creating a much healthier food system.
This is our first science song. This is our only science song of the evening.
Kissing on the corner, and I wonder, should I warn them of the devastating things that could ensue? With these ugly lovers active and the children unattractive, it's a fact the human race can only lose. Are these ugly lovers hoping that the world would go on coping with the kind of offspring only they produce? Though these ugly people know they could destroy the status quo by just allowing any children to be born. If they only could be tutored into maybe being neutered, they could easily avoid the public scorn. But I think that I should mention there's a science of prevention with intentions we should ultimately learn. And in science fun to wish that in some lonely petri dish there is a lovely child waiting for a turn. Splitting the genes, we'll all go splitting the genes, we'll all go together when we go. If the parents need adoption and they want another option, there's a lot of things that they can now explore. As they're thumbing through the file, they can choose the kind of smile that the child could have never had before. If the scientists are friendly, then the money that they're spending will be ending any worries for a while. They can choose 11 toes, or if they want a bigger nose, or just propose a child run a faster mile. Splitting the genes, we'll all go splitting the genes, we'll all go together when we go. All the ugliest of people, how they patiently have waited for the doctors to discover how to fix what has been faded, for the scientists to show that they are truly dedicated, trying to recreate what the creator has created. If the features are recessive and the parents are obsessive, then the guesswork can be easily replaced. If the chromosomes are sorted and then magically transported, will it change the sense of order to the face? If the creatures have been altered and the doctors haven't faltered, can the vault be opened up for all to buy? Cause it has to be enticing to employ a little splicing, thereby slicing out a piece of human pie. splitting the genes, splitting the genes. We'll all go splitting the genes, splitting the genes. We'll all go together when we go. Splitting the genes, splitting the genes. Splitting the genes, splitting the genes. We'll all go together when we go.